All right. Hey, everybody. Welcome back to the Animals 2 lab. Today, we're going to be doing activity 2, focusing on the phylum Chordata, specifically looking at the non-vertebrate chordates. So remember that be sure to watch this lecture, utilize the materials that we provide to you all, whether it be videos or additional reading to help you understand the material, and then be sure to complete the associated questions with this material on your worksheets. But with that, let's go ahead and get started. So in the previous video, we talked about the phylum Echinodermata within the deuterostomes. Remember that there were 4 phylum within deuterostomes, but for this lab, we're going to focus specifically on the phylum Chordata. And so the phylum Chordata, also known as chordates, exhibit bilateral symmetry. What we're going to focus on are these 4 key characteristics that chordates have during embryonic development. So at some point during development, all chordates exhibit these 4 characteristics: having the presence of a notochord, a hollow dorsal nerve cord, they will have either pharyngeal slits or sacs, and they have a post-anal tail. So within the phylum Chordata, there are also 3 different subphyla. We have Cephalochordata, which are your lanceolate, Tunicata, which are your salps and sea squirts, and then finally we have Vertebrata, which is our broadest subphyla, which includes mammals, fish, amphibians, reptiles, and birds. So a variety of different organisms within our final subphyla. But for this section of the lab, we are going to focus primarily on Cephalochordata and Urochordata. But do note that chordates encompass a variety of different organisms, from reptiles to amphibians, mammals like us, as well as a variety of different fish species. All right, so going back to just general characteristics of organisms in the phylum Chordata, these illustrations are just showing that 4 different chordate embryonic characteristics that at some point during embryonic development, all chordates exhibit. But for some species, all 4 of these characteristics are retained into adulthood. But for other species, some of these characteristics are lost from embryonic development into adulthood. So if we look at cephalochordates, for example, we can see that in an adult individual, this organism exhibits all 4 of our chordate embryonic characteristics. It has the presence of these pharyngeal slits, which is illustrated in this yellow color. We also have the presence of a dorsal hollow nerve cord, which is this blue line running the extent of the body of this organism. We also have the presence of a notochord, which is directly below the dorsal hollow nerve cord, and it's illustrated in red. And then finally, we have the presence of a muscular post-anal tail, which is shown in this kind of dark brown color. And as the name implies, it is a tail behind the anal hole of the organism. And we can see that within urochordates, that during development, all 4 of these characteristics are still present using the same color-coded system. We have a dorsal hollow nerve cord, we have a notochord, we have the presence of these pharyngeal pouches in this case, and we have a muscular post-anal tail. But as I mentioned before, not all organisms retain all 4 of these chordate characteristics into their adulthood. And we can see that in an adult urochordate, that the only chordate embryonic characteristic that is retained is the presence of this pharyngeal pouch within the organism. And that's also true for vertebrates. You can think of us that we have pharyngeal slits during development, but they don't turn into gills. We don't have gills as adults. So yeah, depending on the organism, not all of these chordate embryonic characteristics are retained into adulthood, but they are present in our developing embryo at some point during development. But we'll talk about more of that in some of the other labs. All right, so let's go ahead and take a look at the subphylum Cephalochordata. And this refers to what we commonly call lancelets. Basically, these organisms are marine, fish-like in morphology, meaning that they're found in saltwater ecosystems. They're not found in freshwater or terrestrial places. And even though they look similar to fish, they're not actually fish. But what's interesting about Cephalochordata is that they retain all four of the characteristics exhibited during chordate embryonic development. All four of those are retained into adulthood for this organism, which makes them an excellent model organism to look at chordate development because we can see how these characteristics are developed over time during embryonic development. But these guys are pretty interesting in that they are filter feeders. And so what they'll do is burrow part of their bodies into the ocean floor and expose their mouth parts in order to filter feed. So basically, if you're looking at the lancelet here, you can see that its mouth is full of these cilia, which familiar word if we think back to our bacterial labs. They have these cilia-like mouthpieces, and this allows them to pull basically food out of the water column and move that through their bodies. And basically, they're going to filter feed by using the cilia, pulling food into their bodies and passing it through these pharyngeal slits in order to properly filter feed. So this is just a picture of what they look like when they're burrowed into the ocean floor. And yeah, they're pretty interesting little fish-like non-fish chordates. All right. So, as I mentioned before, Cephalochordata, our lancelets here, they retain all four of our chordate embryonic characteristics. So they do have the presence of a notochord, which in the Cephalochordata body type, it stiffens and provides support for the animal. Whereas in vertebrates, this notochord will eventually become a true backbone or a vertebrae. The postanal tail, as we can see, is at behind the anal portion of the body and is basically retained to help the organism move around. So it's used for locomotion. And if we compare that to what this postanal tail becomes in vertebrates, for some species, the tail is retained, if you think about like a dog, whereas for other species, it is not, because you and I don't walk around with our tails present, at least I hope. But all right, so we also have pharyngeal slits in Cephalochordata. And as I was mentioning before, this helps lancelets filter feed. So they also are responsible for mucus production, but ultimately they're used for filter feeding for the lancelet. In vertebrates, these slits become gills for aquatic species like fish, or they further develop into mouth or head parts, because again, we don't walk around with gills on our necks. But, all right. And then finally, we have the hollow dorsal nerve cord. And this is, again, part of the nervous system. And similarly, in vertebrates, this will also become part of the nervous system in true vertebrates, with that developing into the spinal cord. All right. So this is kind of one of the subphyla we're going to talk about in our non-vertebrate chordates. And the other are in the subphylum Tunicata, or the Urochordate lineage. So these organisms are planktonic, and they're basically encased in like this tunic-like structure, and they're often referred to as tunicates. The organisms are encased by a protective tunic, and this tunic is comprised of polysaccharides. And this external tunic basically helps provide support and some form of protection for the organism. And as we talked about in one of the earlier slides, within Tunicata, again, during the early development of this organism, they do exhibit all four of our chordate embryonic characteristics, but as they continue to develop into adults, the only part that they actually retain are these pharyngeal slits into adulthood. So we can see that within the internal structure of these tunicates, that they have all these pharyngeal slits. Just like the cephalochordates, tunicata, they are also filter feeders, and basically they eat by pulling in food with these incurrent siphons. So they're like siphoning water into their bodies and like filtering out food through their pharyngeal slits. And then they're passing that out through an X current siphon. And again, they're basically flushing water through their body and filtering food out of that water in order to eat. But really, that's all we're going to talk about for the non-vertebrate chordates. If you have any questions, feel free to email me or your own lab instructor, and I will see you in the next video.